Hi, this is Miss Litton, and it is Friday afternoon, and these are my wonderful AP biologists. Say hi.
Hi.
And we're here on this beautiful sunny Friday afternoon to review Chapter 4, which is our introduction to prokaryotic and eukaryotic cells. So let's hit the main point, shall we? First thing we needed to review real quick is the cell theory. And you're going to know the parts of this cell theory because you're going to know that all living things are what?
Organized.
And that's really good. And made of cells. Yes. Okay. And that cells are the what?
Structural and functional unit of organisms.
That's not rude. And 3, new cells only come from pre-existing cells. That is the cell theory. And when we look at the scale of cells, the main point I want you to get here is that cells are very, very, very small. Who is smaller? A prokaryotic cell or a eukaryotic cell?
Prokaryotic.
Who has more surface area to volume? Right. Prokaryotes do because they're smaller. Who's smaller than a cell?
An atom.
You're right.
Organelles.
Okay, yeah, organelles, right? Viruses, right? Those are smaller. What do we look at small things with? We look at them with what? Microscopes. So light microscopes, you know, maybe we can magnify up to like 1,000, 2,000 with a light microscope, which is what we have here in the room. But even better still would be a what? Electron microscope. Okay, that's all I wanted you to know about that. I also wanted you to make sure you understood between magnification and resolution. Magnification is how many times you what? Increase the size. Good. How big is it? And resolution is how clear it is. And then there was something we learned a little bit about when we talked. Interesting. Okay. Then the other thing we talked about was, I hope it doesn't stop recording. Somebody remember to send me a text that I need to do that or something. Was field of view. The more we magnify an object, what? Smaller our field of view. That's what we're seeing of it, right? Just like it sounds. Field of view. What am I viewing? If it's less magnified, I'm seeing more of it, right? A larger field of view. If I'm magnified, I'm seeing smaller. Okay, that leads us up to prokaryotes. Let's think about some things we know about prokaryotes. Let's start from the outside and work our way in. What is the outermost layer of a prokaryote called? No. Starts with a G. Outermost. Outer, outer, outer. Glycocalyx, right? We have a capsule. Remember that on the outermost part of it? The capsule. Outer, outer, outer. Outermost layer, right? And what can be attached to that outermost layer? No. Yeah, okay. So it's called fimbriae, and it helps it attach. Fimbriae helps it attach, or what? Flagella. What else could I be extending out of my outermost part? It will start to come out of it. A what? A sax pilus. That helps with conjugation. So maybe let's go back. Let's go. Oh, look, there's some bacteria. Oh, look, there's a cell membrane. Here we go. Okay, this is the hairy Horndog part. This is what I'm talking about, hairy Horndog. Okay? So you have these carbohydrates on the outside. That's the hairy Horndog part. That's the carbohydrates. And all these little tiny, looks like little wispy things, those are the fimbria, that help it attach. Okay? And then you could have the flagellum, which helps it what? Move. Good. And you might see coming out of that a sax pilus for conjugation. You use that to pass your DNA. Now, if we go in one layer from that, I'm working backwards from the notes, right? Because in the notes I went membrane, right? Now we're going backwards, outside to in. So next layer in would be what? Cell wall. Good. That's your next layer in. Remember we talked about peptidoglycan? It's not made out of the same substances as you would see in a plant cell wall. It's a different substance. Okay, so now we're at the cell wall. What's the point of the cell wall? Separate, structure, support, the shape of the cell, right? Okay, let's go in one layer from the cell wall. What's the next layer in? Cell membrane. Definitely not a Golgi apparatus. You know why? Because this is a prokaryotic cell and it doesn't have any organelles, right? Okay, so the next layer in would be our cell membrane. Okay, now this is where it was just a really good example for you to stop and think. Because in your notes, I listed cell membrane, cell wall, glycocalyx. Here's the outermost layers, right? But I did from the innermost outer layer to the outermost outer layer. Did you follow me on that? This time I went the other way, and some of you had a hard time with that. You said the cell membrane was on the outermost side. Okay, so listen to the question that gets asked so you get the right answer. You got to think it through. Okay? So, yes, you're right. If I was starting from the inside, it would go cell membrane, cell wall, glycocalyx. But I said for you to start from the outside, glycocalyx, cell wall, cell membrane. Check with your bio buddy and make sure they get that right now. Ask them. Okay, now let's keep going in. Okay, so inside the cell, we don't have a nucleus, we don't have Golgi apparatus, we don't have endoplasmic reticulum. Why? We are a what cell? Prokaryotic cell. So we don't have that by definition. We have no membrane enclosed organelles. Well, I do see things in there. I see things like a dark, big region in there. And what do you think that is, that darker, staining region inside? Tell me, everybody. Nucleoid. Yeah, the nucleoid region. And what's inside the nucleoid region? No. DNA. The nucleoid region is where the DNA is located. What does nucleoid sound like? There's something else you know. It sounds something similar. Nucleus. That is the membrane, right? Membrane enclosed DNA inside of a eukaryotic cell. And inside the nucleus, you could have a nucleolus, and that is where the ribosomes are made, the rRNA for the ribosomes. But that's in a eukaryotic cell, right? So in your head, this is what you need to be thinking right now. In your head, you would need to think there's cells, and I have the cell theory. My cell theory says what? All living things are made of cells. And that those cells are the structural and functional units of all living things, and that cells only come from pre-existing cells. What else do I know about cells? Cells are very small. Why do I need small cells? Because I need a large to volume, right? The third thing I need to know is there are two general types of cells. What are the two general types of cells? Prokaryotic and eukaryotic. What's the difference? Prokaryotic cells have no membrane enclosed organelles. This is one right here. It's a prokaryotic cell. Eukaryotic cells are bigger, larger, and they do have membrane enclosed organelles. Hang on one second. Hi. Your haircut looks nice.
Thank you.
My theory is actually just pausing you for a second. Okay, so come back to me. Prokaryotic cells have no what? Membrane enclosed organelles. They have DNA, but it's not inside of a nucleus like us. It's just called the nucleoid. They do have workbenches for protein synthesis. What are those called? Ribosomes. Good. So they have ribosomes. So that's also in there. They also have little extra pieces of DNA, little extra rings of DNA. Do you remember what those are called? Starts with a P. You're going to genetically engineer them. Plasmids. We're really glad our test isn't Monday, okay? Plasmids. All right, so then that brings us to here, okay? So there are 3 domains. All life can be broken down into 3 domains. Eukaryotes like us, right? Prokaryotes are into 2 categories, bacteria, which we just discussed, and archaea. What's the difference between bacteria and archaea? Cell walls are a little different, cell membranes are a little bit different, and archaea are what's called extremophiles. They live in extreme environments. Okay, okay. Now, this brings us to eukaryotic cells. And the first thing I might ask you is,
I texted it too.
thank you so much. I super appreciate it. You're awesome.
Let me know if it doesn't work.
Okay, thank you. Eukaryotic cells, this is not part of our notes, but part of it is because I talk about differences between our energy-related organelles. Do you remember that part? It's in the notes, that part. And that is, how could a eukaryotic cell have evolved from a prokaryotic cell? That's because one prokaryote went and lived in another prokaryote. Who would that be that went and lived in another prokaryote? Mitochondria and what? Chloroplast. Our evidence, which is in your notes, is the mitochondria and the chloroplast have how many membranes? 2, because maybe they got engulfed. They have their own, and it's in a ring, not in a nucleus, in a ring, just like it would be if it was a bacterial cell. They have their own workbenches for protein synthesis. What do they have? Ribosomes, and they are self-replicating. Okay, so because your energy-related organelles can do that, maybe they were once independent, free-living prokaryotes that went to live in another prokaryote. Yes?
Is there a name for that theory?
Yes, the endosymbiotic hypothesis, which I thought I was on, but apparently I was not. Endosymbiotic hypothesis. Okay, then I told you, you need to know the structures and the functions of all the different organelles. You have probably in your notes pictures of them labeled and what they do. You have a quizzes, right, that I gave you to learn the parts. But if we looked right here, if I asked you, is this an animal cell or a plant cell? How do you know that? There's no chloroplast. What else? No, there's a nucleus, and they both have nucleus. No cell wall. There's not a large central vacuole. There are vacuoles, but not a large central vacuole. What else? The plant and animal cells have mitochondria. If you're a eukaryotic cell, you have mitochondria. It's just that plant cells have chloroplast. What? They both have that. There's something an animal cell has that a plant cell doesn't. Centrioles, the churros. Because only animals eat churros. Everybody knows that. Okay? Only animals eat churros. Here are the churros. Okay? So now, again, look here. Animal cell and plant cell. Plant cell has a cell wall. It has what? Chloroplast. It has a large central vacuole. It does not have what? Centrioles. It doesn't have the churros. See the difference? Okay. If I asked you, guys, if I asked you who the doorman was, who decides who goes in and out of that cell, what would you say? Who goes in and out, it would be the what? Cell membrane. I agree. I don't know where the cell membrane is on this picture, but I agree with you. Somewhere. Okay? Who would you say detoxifies? Lysosomes might do it. Some smooth ER might do it. Peroxisomes might do it. Who supports? No cell wall here. Cytoskeleton supports. Good. Who's the UPS man? Golgi body. There he is. Ooh, what's the side facing the nucleus? The cis. And the one going away is the trans. Good. Who's for energy? Mitochondria. Good. Who stores? Some vacuoles. I'm sure there's a vacuole on here somewhere. Anyway, a vacuole. That looks like a vacuole there. Who does protein synthesis? No. Ribosomes. You won't tell me ER, because it's not the ER that does the protein synthesis. It's the ribosomes that may or may not be sitting on the ER. Right? Okay? Who has ribosomes? Rough ER. Who's the brain? Nucleus. Who digests? Lysosomes. Okay, that's good. Good on that. Now, I'm going to jump to here. Guys, you have this short window when you're with me. Let's use it wisely. Okay, so don't hang. Okay? This is a good diagram because, yes.
Who detoxifies?
Well, several can detoxify. The smooth ER, peroxisomes can detoxify. I guess a lysosome could digest, but I wouldn't put it underneath the detoxify umbrella. Okay? So here, when I look through this story, you fill in the blanks as I tell the story. Your code book for your cell is called what? DNA. DNA is inside of the nucleus. It will never leave the nucleus. So when it wants to get its message out, it will use what? RNA. What kind of RNA? And it'll make a copy of the gene and it'll leave through the nuclear pore. And that mRNA will go out and sit on a workbench for protein synthesis, which is called a ribosome. When that what does the M stand for? Messenger RNA. It has a message there on how to build proteins. What are proteins built out of? Amino acids. Who will carry the amino acids to the message? tRNA, because T stands for, and it's transferring amino acids. When you have amino acid 1 land, amino acid 2 land, they will join by what process, what reaction? Forming what kind of bond? Good. And I will hook a series of amino acids together based on the messenger RNA, which is sitting on the what again? Ribosome. Ribosome, which is made out of what? What's a ribosome made out of? RNA. Oh, yeah. And proteins. And who made that rRNA?
The nucleolus.
The nucleolus did. Thank you. Okay? Now, I have built a protein. I'm on a ribosome. The protein is built. It will probably move into the, ooh, I need some stitches. ER. And if I'm a ribosome on ER, that ER is called rough ER. Good. So now the protein moves into the ER. It could be modified, changed, and it might move from the rough ER to the smooth ER. But when it leaves the smooth ER, it'll go into the what side? Right, and it'll be further packaged, modified, and you'll put on a address label, and it'll leave on the and now it might go to the edge of the cell and export, or maybe it's the stomach of a cell called a lysosome. Too many classes today. Lysosome, and I might hook up with an endocytotic vesicle. What was that? Endocytosis. Okay. So you ate something via an endocytotic vesicle. Do this. Okay? And then I'm a lysosome, and she ate a Chromebook. Okay? And so I'm going to come up here and I'm going to join, and we're going to do what? Digest. We'll absorb nutrients out of here, and then whatever we don't like, we will go over and we will do exocytosis. Okay? You see how we did all of that? Okay. This summary. I killed you last time? I don't kill anyone.
You did get it last time.
I did. Okay, come back here. It's all on this summary right here. Where else could you find this summary? You can find this summary at the end of the textbook. Where else could you find a summary? At the end of your notes, there's a summary. Okay? Those are all resources for you to use. Okay? Peroxisomes are going to take free hydrogens. They're going to hook them up with some oxygen. We're going to form some hydrogen peroxide, which we're going to eventually break down anyway. Now we're at the, large central vacuole in a plant. But for us, it could be a digestive vacuole, or it could be a vacuole that a protist is going to use in order to excrete water. It could be a storage vacuole. So vacuoles, what I would call them, is the multi-purpose room. They could be used for a play. They could be used for an assembly. It could be used for a classroom. It could be used for lunch in the rain. Okay, it is a multi-purpose kind of organelle that you could use. Now, the energy-related organelles, we know some things about them. They have their own. They have a double membrane. They have their own ribosomes. They are self-replicating. Okay? Right? That's true about both of them. Okay? But chloroplasts in particular are part of a bigger group called what? And more specifically a chromoplast, which means it has what on it? Color. But it is green. Other ones could be used to store starches and oils. Hey, look, there's a chloroplast. Yay, look, there's mitochondria. Now we're at the cytoskeleton. Our cytoskeleton comes in 1, 2, 3 sizes. The smallest size of all is what? And I always remember active actin because it's smaller. Okay, active actin. It's two chains wound together. Actin. Then intermediate filaments. You have several chains wound together. And then microtubules are a little bit larger. All of these work to provide what? Support, structure, transport inside the cell. Okay? And so you can see that we're going to see this tomorrow in class. Okay? I have a little video to show you. Not tomorrow. They're all coming in on Saturday. Okay? You can use, you're almost done. You're doing a great job. You can use microtubules. Here I am. I'm the myth. Microtubules to build things like churros, centrioles. Who has these? Only like what? Plants and protists. Okay. So notice the triplets. How many triplets do we have? 9. This is the 9 plus 0 arrangement. Okay? Now, nothing in the middle. It looks like something's in the middle, but there's not. It's really annoying. Okay? So 9 triplets. That is a centriole. There's 2. They're perpendicular to each other. You see them in animal cells. They divide, and you have 2 of them during animal cell division. Microtubule organizing center in the center of the centrosomes shoot out microtubules to help with cell division. Okay, we're going to learn more about that when we study my arms, my legs, my toes. Exactly. Now, microtubules in this arrangement, 9 groups of 3, are also the, I don't know how to make this, anchor, anchor for flagella and cilia in that same arrangement. But when you look at, and it's not 9 groups of 3, it's 9 groups of 2 with 2 in the middle. But down where they're anchored, it looks the same. So here we can see that 9 groups of 2 and 2 in the middle, and there's a diagram here. 9 groups of 2, 2 in the middle. That's cilia, flagella. That would be centrioles. That would be basal bodies. All right, let's try a little something, something. Actually, before I do that, what is this? Cell membrane. Good. What is this? What is this? What's this? What's this? Nuclear pore of a nucleus. What's this? What's this? Those were what? Centrioles. All right.
Wait, what was that again?
Hi. Okay. Now, what was what? Want to go back? Do it again. Do it again for me. What's this right here? What's this? Perfect. What's this? What's this? What makes it rough? What happens on the ribosomes? Who dictated the protein synthesis? Who got the message out from the DNA out to the ribosome? So smart. What's this? What does that make? RNA. Good. And RNA will be part of the ribosome. Good. When we hook ribosomes together, what's the reaction? What? Do I use water? Do I make water? Make some water. Okay, good. And what's this right here? Good. What's that? What are these? I'm not going to say churros. Centrioles. Okay. Plant. What's this? Yes.
What is the function of a centriole besides to anchor?
You know what? I don't know. No, I mean serious. We don't know. They are found at that microtubule organizing center, which more microtubules come out to help during my arms, my legs. Exactly, but nobody really knows. That's a doctoral thesis. What's this? That helps with what kind of pressure? I didn't review.
Turgor pressure.
Turgor pressure. Boom, shakalaka. What's that? What's that? What does the cell membrane decide? Good. What's this? What's it made out of? What important organic molecule is that? Carbohydrate. Is that for structure or energy? Structure. If it wanted to store energy, what would that be? Starch. Good. What's this? Good. Okay. I'm glad. Okay, let's do this. Go in as your favorite cereal. Favorite cereal. It's going to connect. I don't know why it's not connecting. I have no idea. It's working? Here, we're pausing you. Okay, right now, students are working through the 13 questions I asked, and when we're done, I'll restart so you can see the questions, okay? Okay, we're going to go ahead and stop it. Here we go. Good job. Powerhouse, mitochondria. Stop. This is one that I've seen has this misconception before. They think the cell wall decides what goes in and out. Cell wall is just either peptidoglycan, it's cellulose, it's chitin. It doesn't decide what goes in and out. The going in and out, the dormant, is always the cell membrane. Number 3, processes, packages, distributes, Golgi apparatus. The ER could modify and send, but when you say process, package, and distribute, you're mailing, it's got to be the Golgi. Somebody said lysosome. No, you're just wrong. Which organelle is the stomach? Now you want to say the lysosome. 4 of you said ER. No, you do not digest in there. Not the Golgi apparatus. Stomach is the lysosome. Support. Protects. Permeable to gases. That is going to be your cell wall. Protects, supports, permeable to gases. That's your cell wall. Not your cell membrane. I don't think about the cell membrane as protecting, but a wall does. Not one between California and Mexico. Yeah. Oh, I saw a hand up. No, Eddie does not need me now. Not vacuoles. Vacuole can support a plant cell, but not the other. Okay, don't talk. People are trying to hear. Everybody got this right. I would hope so. ER is rough if it has ribosomes. Chloroplasts aren't rough. What's wrong with you? Converts light energy to carbohydrates. Definitely a chloroplast. Not mitochondria. Lysosome? No. Stores, transport, support, yes, vacuoles. Seriously? I'm not even going to address those. Which structure is the site of protein synthesis? Ribosomes. Ribosomes. Which of the following structures will be found in both plant and animal cells? Yes, mitochondria. 2 people said all of these are found in both cell types. Centrioles only in animals. Chloroplasts only in plants. Cell wall only in plants. Okay. Which of the following has 9 sets of 3 microtubules? Centrioles and basal bodies. What do basal bodies do? Anchor, cilia, and flagella. Yes. Are you raising a hand, baby? Okay. Which of the following has 9 sets of 2 microtubules, 2 in the middle, just cilia and flagella? Centrioles are 9 triplets. Basal bodies are 9 triplets acted as a single protein. What's wrong with you? Cell membrane? No. No. And that's it on that. Okay? Review, review, review. Look over your essays that are appropriate. You have old school and new school. If you missed the essay review, you should watch it. Okay? Watch it. And then look at the questions at the back of the chapter. And you're going to be great. And we'll talk about Chapter 5 on Monday. I love you very much. Have good weekends. No sex, no drugs, no alcohol. What happens in Vegas? Thank you. If you get hungry, have a piece of toast. I love you.